basement membrane, and under it there is the lamina propria. Stratified means the cells are layer by layer, one layer to another one. As this, for example, as apartment, yes, there are some houses located to under them, but the simple epithelium is just the house that have a yard or garage or other things. The stratified epithelium, the basal layer located on the basement membrane, and the other one located on it that have maybe 2 layers or more. This is the division based of the layer of the cells. This cell is epithelium, simple or stratified, again based of the shape of the cell, divided to 3 types: squamous, cuboidal, columnar. Under microscope, as I said for you yesterday, we can see mostly the nucleus. And for identification of the cell type, we should notice to the shape, nucleus shape. Squamous cells are flat cells. The height of the cells is shorter than its base, as this is stone. Yes, the cells are flat. Squamous means the flat stone cell. The nucleus of the cell is as same as to minus sign, a line, a narrow line. Squamous cells, as this. The second one is the cuboidal. The height and width of the cells is equal. The nucleus is round shape, cuboidal shape. The third type is the columnar cells. The height of the cells are more than its width, as this, cuboidal, or maybe no, they are as round, as a columnar shape, yes? The nucleus have oval shape. Yes? The shape of the nucleus is important because we can see under microscope the nucleus better. Simple squamous, one layer, simple one layer. Simple squamous, one layer of the squamous cells, just one layer of these cells that line the vessels, and also some membranous structures that we know them as peritoneum, pleura, and the pericardium. They are the serous membrane. The name of the simple squamous epithelium in the vessels named endothelium, in the serous named as mesothelium. These tapers are important, yes, for example. And also you should know, because in the other semester that you know about the systems, because Ahmed said to me that there's some important things, and also I said to you some questions or the sample of the question that you know which part is important you know. The book of the Jambuira have lots of knowledge that all of them is not necessary for you. It's better you know, yes, but not necessary for you. For my session, yes, there is slides, the PDF, and also my explanation enough for you, for your exam. And also, I said to the Ahmed, we write a book that summarizes the Jambuira as essential histology. That I said to the library that buy and have here, but I don't know, they bought in the expression there or not, yes? This book also is okay, but for your exam, yes, because most of the cell and the tissue development, the histology is for me, just the cartilage and bone is for Dr. Milan. For my session, this PDF enough for your exam. And most of them, around 80% of them also is enough for your basic science exam. Maybe around 20 to 25 questions are not. Because the Jankuira book is included a little of details, a little of molecular subjects that are not necessary for you. They are for the specialist. This is one. This is important. The type of the epithelium and their location. We don't need to memorize the function because the function related to the physiology. Simple cuboidal. One layer of the cuboidal shaped cell are in the thyroid follicles and also covering of the outer surface of the ovary and also some growing follicles of the ovary in the cycle in the woman. For example, the primary follicles also lined by simple cuboidal. Simple columnar, one layer of columnar cells are in the GI system from the stomach, small intestine, large intestine, and upper part of the rectum, gallbladder, and also in the uterus, lined by the simple cuboidal. The shape of them under microscope. The squamous shaped cell. Yes, they are very flat and narrow and thin cells. Under microscope, their nucleus is as same as 2 minus sign. There as a very thin line, perpendicular line. Cuboidal. Round nucleus is that I show in the pointer. Show the round nucleus. They are just in one layer. This picture shows the thyroid follicles that in the practical we see them and now them. Columnar. The height of the cells are more and the shape of the nucleus have the oval shape. Or here have oval shape that we can identify. One layer of the cell, simple, columnar shape, oval nucleus. Stratified epithelium. More than one layer. 2 or more. Maybe 2 or until the 50 or more layers are in some parts. Stratified. They have again some types. Squamous, cuboidal, transitional, columnar. But then they have different layers. How we name them as they shape. They are layer by layer. The outermost layer shape used for the naming. If the outermost layer have a squamous shape, the type of the epithelium is a squamous stratified epithelium. Again, if in the end of the squamous cells included the keratin filaments named as keratinized squamous stratified. The keratinized are in the dry parts, in the skin just. Non-keratinized are in the wet structures, in the oral cavity, first part of the nasal cavity, esophagus, in the larynx, in the vagina, and in the anal canal, lined by the squamous non-keratinized. Outermost layer included the squamous shape. If the outermost layer have a cuboidal shape, layer by layer, the outermost layer have cuboidal shape, named as stratified cuboidal epithelium, are in the sweet gland and also developing ovarian follicles. There are lots of keratin fibers are keratinized squamous stratified in the skin. Stratified cuboidal. Stratified more than one layer, for example, here 2, here 3 layers. The outermost layer have cuboidal shape. Outermost have round nucleus, show the cuboidal shape. In the transitional epithelium, the cell also are layer by layer, but the shape of the cell when the organ full or empty is different. Urinary bladder. When the urinary bladder is full of the urine, yes, the wall of it is stretched. The shape of the cells is very narrow. But when it's empty, yes, the cells are very big. Because of it, we name this type as a transitional epithelium. We can see this type of epithelium just in urinary system, also named as urothelium, epithelial tissue of urinary. In the urethra, urinary bladder, ureter, and also the calyx of the kidney. You can see this type of the epithelium. This type of epithelium have one specific cells that the shape of the cells. Yes, here. They are very full. Yes. The cells are narrow. And under it there is a very big cell. But when they are empty, cuboidal shape, basal cell, and included some big cells as dome shape, and again in the outermost layer, in both of them there is a very big cell. These cells cover more than one cells located on some cells as an umbrella. Umbrella, yes, we put it, maybe some person stand under it, 2, 1 or more, yes. These cells, because cover more than one cell, named as umbrella cells that are just in the transitional epithelium, or for example in urinary bladder. Maybe one of these questions, where is the umbrella cell? Maybe the answers be different type of epithelium, for example pseudostratified, transitional epithelium, squamous or other one, or no, maybe are in the organs, for example in the thoraca, in the urinary bladder, in the stomach or others. Yes, the type of the epithelium have 1 or 2 questions in the exam. So the stratified epithelium, yes, compared to this one, yes, here cells are different layer, but here the cells are just all of them in one layer, and the nucleus are in different layer, and in the apical also we can see the cilia, because just this type of the epithelium are just more in the respiratory system. Different type of the epithelium, the schematic picture and also the location. The other type of the epithelium is glandular epithelium. Glands. The glands that we know them as exocrine glands and also endocrine glands. Why they are the type of the epithelium? Because as you see here, in the embryonic period, they derive from epithelial cells. The epithelial cells proliferated, increased their number, invaginate to under connective tissue, and finally differentiate to the glands as exocrine or endocrine. The exocrine have duct, the endocrine don't have any duct, they are ductless. This part is have another system. We have one system in our body as endocrine system. that you now in the future in the endocrine system. But the exocrine glands are spaced in different systems. For example, the sweat glands in the skin, the salivary glands in the GI system, the pancreas, the liver, yes? Some glands as prostate in the genital system. All of these are exocrine glands. These glands have some division. This is the difference of them. The first division is based on their number. Yes, the exocrine glands have some classification. The first one, based on the number of the cell in each gland. Unicellular, multicellular. We have just one gland that have just one cell, the goblet cell, that are in different covering tissue. For example, in the respiratory system, in the GI system, in the reproductive system, included just one cell, one large, very big cell that secreted the milk. But another one, salivary glands, sweet glands, or other one are multicellular. They have more cells. Yes, this is one classification. The second one is based on the shape of the cells. The shape of the secretory portion or the duct that have the table in your book. Just a study for yourself, because you should know this and it is useful for you in the future, in the other system. The third one is about the model of the secretion, how they release their product. First one, by exocytosis. Yesterday we talked about that. The cells release their product by exocytosis. The secretory vesicles accumulated in the apical part, and then the secretory vesicle released from the cell, such as sweat gland and salivary gland. Apocrine gland. Additional to its product, the apical part also removed. Apoapex, yes? Memorize it. Apoapex. Apocrine apex also released, are in the breast for the secretion of the milk. Holocrine. Hole of the cell ruptured and released. Or in the sebaceous gland, in the skin, or this example. As you see here, the cells at first divided to some small parts, and finally all of the cells released. This classification is of the base of the model of the secretion. The other one, the exocrine glands product, the type of their products. They produce seros or mucus. Do you know the difference between them? The seros included more proteins. Both of them included the proteins and carbohydrates. The lipoprotein or protoglycan, that we now got difference of them in the connective tissue. Included both of these. The seros have more protein and also more water. It's bodily and used mostly for washing of different parts. For example, around the taste buds, around the olfactory epithelial, there are some serous glands that release their product for washing of the taste bud. Today you notice that when you eat some things, yes, you can taste all of their taste. For example, the salt, the sour, or I don't know, you eat the kebab or other things, yes. When you eat each one, yes, you can taste it. Why? Because immediately after the eating of food, the serous gland wash the taste bud, and we can again taste another bud. But the milk similar to the glue, yes, attached to our hands or others. This is the difference between these two glands. The serous mucus, based on their secretion, their organelles, and also their picture on their microscope. Serous glands included serous cells. Serous cells have pyramidal shape with round nucleus and also basophil cytoplasm. Everywhere that included more proteins, more enzyme under microscope have purple color. Yes, the nucleus have purple color. Why? Because they included the ribosome, they included the RNA that are basophil structure. They like the hematoxin. And you now, yesterday again I explained that RER, Golgi apparatus, involved in the protein synthesis. They have a well-developed ostis. Serous glands included mucus cells that have more carbohydrate. Under microscope, when we prepared a slice that yesterday I explained about that, we use the alcohol and xylene, yes? Remember that, yes? Alcohol and xylene wash the carbohydrate, have more carbohydrate. Wash the cytoplasm, the granules that are in the cytoplasm. Because of it, under microscope, the mucus cells are poor stain, have white cytoplasm, and these granules push the nucleus to basal part, have basal flat nucleus. The serous cells, difference between of them. Serous cells have basophil round nucleus close to the basal part, not in the bottom of the cells, and this basophil have purple color. The mucus cells have white cytoplasm, poor stain, and flat nucleus that is attached to the basal surface, located in the peripheral part of the cells. That we can see them in the salivary glands, in the cerebral cells that are in the respiratory system, that you can see them, and we won't see them in these cells, that just you know about them. In the first slide, I explained for you that there are one of the function of the epithelial tissue is included some cells as myoepithelial and this contraction. What is the myoepithelial? Myo means muscle. It's another word for the muscle. This is finished. Yes, the exocrine glands have 4 classifications based on different subjects. Myo means muscle. Myoepithelial are some cells that derive from epithelial cells in the embryonic period but act as a muscle tissue, act as contraction. They included the actin and myosin filaments, they containing myosin and a large number of the actin that they interact together and contraction. Where they located around the glands. Yes, this is the glands or duct. I don't know, maybe sweet gland, for example, or their duct. Located around them, they have flat nucleus, electron microscope. They have a lots of cytoplasmic process that surrounded the nucleus. When they contract, push to the glands, facilitate the secretion of the glands, exit of the products. They are myoepithelial cells, are derived from epithelial cells, act as a muscle cell, have actin and myosin, and they function with contraction around the secretory or conducting portion that help the secrete the products. And again, in the first slide, I explain for you that the epithelial tissue have some cells as basal cell. These basal cells are in all part of the epithelial tissue, in all part of the body that lined or covered by epithelium. These cells are stem cells. Do mitosis, gradually renew itself, and also they also can involve in the regeneration when the epithelial tissue damaged. And this renewal occurs in the epithelial tissue. This is the chapter 4 of the Jankuira about the epithelial tissue. For the epithelial tissue, what you should, I'm sorry, you should study all of it. Explain a little about the tissue development exam. I think around 10 chapters. The epithelial tissue, connective tissue, blood, bone, cartilage, muscle tissue, nervous tissue, and the skin. Yes, nervous tissue have 2 sessions. I think 10. In each session have 4 questions. That we divided that the questions are equal in all of their topic and subject. Yes. For example, the epithelial tissue have 4 questions. That this question mostly, generally, have 1 question about the type of the epithelium that I example for you. For example, this type of epithelium is there in the urinary system, in the stomach, or other. One of them is about the model of the secretion, is exocrine glands, and also the junctions is very important. You know the different type of the junction. There is main molecules that are in there, and also their function and the location of them. They are, for example, in the lateral surface, in the border. If they are more, for example, the tight junction are more in the barrier, are more in the urinary system. The cell is the separate. Yes, you have a separate exam for the cell and molecule. That I think have 9 to 10 questions for the cell for my topics. For the survey topics. And the more of them are in the biochemistry that I think is starting the next week. Yes? I think 2 or 3 non-marks is for the cell, and the other 15 number is for the I think around 15 is for biochemistry, and also you have, I think, around 2 sessions about the physiology of the cell membrane. Okay? Thus you should, it's hard, and now this is your first semester. The medicine major is very difficult, especially the basic science, because it included just a lot of knowledge that you should memorize all of them. Yes? And the Iranian students are the same as you, yes? You, don't worry about that. Also, they have this problem in the first semester, because I think now I maybe teach around maybe 30 pages in the book, yes? Because in the first semester, but again, your parents is very lower than the Iranian students. They have also the anatomy in the first semester. Because of it, a little you should try more. You should study a little harder, yes, in the first, because I know your topics, because most of you are late, and the semester start very late, and we should finish all of them, I think, around 2 months. And also because we come from Tehran, again your classes compact. That, for example, we come here for maybe 2 or 3 times in each semester. Because of it, I recommend to you if try that study gradually, yes, not collected there for the exam tonight, because they are study more. What time is 10?
Our examination started 19th of July, right?
I don't remember. Yeah, yes, I know, but I wrote in my calendar. June or July? July. And I think your practical also is close to your theory. I think maybe your exam Saturday, yes? I don't know. I think maybe the international office arranges for you a review of we have a review for practical. This is general, usually we have. But because some of the students late, and I think the next time I come, again, some students are late. Maybe for the newer students, we have a revision for theory, but I don't know. In the next time that I come for the muscle tissue and skin, that just these two topics remain for histology, I don't know. Mr. Naravi should arrange with you and this help me. Maybe you have a revision. If you have revision, please study there. This is close to the theory exam. That when I come, I just explain for you that the part that you have problem for understanding of them or some part of the people. Yes, we start when 10:15 or 10:30. 15.
You have a pressure class today?
No. From today, we start then another block as tissue development and function. That in this block, we have about the microscopic structures of the different tissue that are prototyped as epithelial tissues, connective tissue, and muscular and skeletal tissue. Connective tissue have again some special type that today I explained about the proper and adipose tissue, the cartilage, bone, and blood. But today we have two topics as epithelial tissue, then proper connective tissue, and adipose tissue. And the subthalamic nervous tissue and blood tissue part is for the urinary and the other time that I come, we have two subjects about the muscle tissue, that the tissue finish, and just one organ that because not located in any system also is in the tissue development as a skin. What is the epithelial tissue? Do you have any information about it?
We covered all the body, like skin,
Yes.
and some parts of, for example, people got hurricanes and legs.
Yes?
It is only places where acid transportation is fixed.
Please louder and louder, please, because I cannot hear you.
It is mostly found in places where acid transportation is fixed.
Yes?
Diffusion.
It's the function. Yes?
Lies the
Lines there?
glands of the body
Glands.
performs some protective functions.
It's better that we said that the glands is a type of epithelial tissue. That they explain for you later, and you know why. The glands also are a type of the epithelial tissue. Yes, as you know, Epithelial tissue, as it states, epithelial means above, and epithelium means covering, and this type of tissue cover all of our body as an epidermis layer of the skin. And also, there are some organs inside of our body that have a lumen or a cavity. All of the organs that have a lumen, such as all of the organs in GI system, oral cavity, esophagus, stomach, intestine, or the trachea, yes, all of these have a lumen. All of these lumens also lined by the epithelial tissue. That today we know about the characteristic of the epithelial tissue. It means we have 4 types of the tissue in the body, and each of these tissue have a special cells with a special structure that we can identify them from together. Today we want to know the epithelial cells characterization and how we can identify them from the, for example, muscle tissue, nervous tissue, or other. In the 4 types of the tissues, we should know about the cells and extracellular matrix. Yes, when we study or we want to learn about the tissue, we should know the cells of each tissue and also the type and amounts of the extracellular matrix. That, as you see here, epithelial tissue included some aggregated polyhedral cells. The most part of the epithelial tissue included cells. The cells located very compact, close together, and the ECM is very red. That cover the outer part of the body and also lining the body cavities. The connective tissue have several types of the cells compared to other tissues. I mean, for example, the epithelial tissue have just one cell, epithelial cells or muscle cell, nerve cells. But in the connective tissue, there are a lot of cells that some of them are just and always in the connective tissue, and some of them migrate from the blood to connective tissue and do their function that we know in the next session. Muscular cells in the embryonic period a little elongated compared to other tissues, and the nervous tissue have a lot of process. Connective tissue have more ECM compared to other tissue and have different function. But now we want to know about the first one. The main function of this tissue is covering, lining. Covering mostly used for the epidermis of the skin, cover our body, and lining used for the hollow organs, line the, for example, trachea, line the esophagus. Additional to this lining, yes, the cells that line one part, yes, line this part. But maybe these cells themselves, or no, between of them are some cells that have additional function in their epithelial tissue. For example, you have small intestine cells, yes? What is the main function of the small intestine cells? Absorption of the nutrient from food. Yes, their name is enterocyte. They are in the intestine, and also name them as absorptive cells. Yes, all of the cells line the small intestine but involved in the absorption. Some of them involved in the secretory cell, and they act as secretory cells, such as the glands, that they are one type of the epithelial tissue, or the goblet cells that are spared, for example, in the respiratory system, secreted the mucus, or in the stomach. There are some cells that, for example, secreted the chloric acid, secreted some proteins that involve in the digestion. Contractility that included some special cells that I explain for you later. They can act as some specialized sensory cells, such as tongue. Yes, tongue lined by one type of epithelial tissue, but included some cells that involve in the tasting. The taste bud is a type of epithelial tissue, but act as a neuron, neuroepithelial cells, or in the olfactory epithelium, in the smelling. And finally, all of these epithelial tissue with this function act as a selective barrier that can facilitate or inhibit passing of anything from the skin or other character. The epithelial cells have some characterization that we can identify them from other cells. The first one is the cellularity. It means they included a lot of cells that bound tightly together and included a little amount of the extracellular matrix. Thus, we can tell that the epithelial tissue included more and more cells, and the ECM is very rare, that these cells located neither together tightly, densely, and in the epithelial tissue we can find lots of cells. Vascularity. Vascularity means the vessels. A is a minus it. A in the medicine terminology means the absence. Avascularity means that the epithelial tissue don't receive vessel directly. They give their nutrient from the lumen that line in, and the second one, this is a rule, this is a fact that under the epithelial tissue, in all parts of our body, there is a type of connective tissue named as lamina propria. It's a type of connective tissue, as the proper connective tissue, because they don't receive the vessels. The vessels are in the connective tissue, and these cells receive their nutrient by diffusion or from lumen. For example, the enterocyte in the small intestine, the cells of the stomach, because these cavities are pulling the food. They can also receive their nutrient from these lumens. Attachment. They attach together tightly. They don't separate from together. When this attachment of the cells damaged, resulted to some disease in some part that explain about it, that are in this surface included some junctions, some molecules that attach them together and also to the basement membrane. They are rich in the nerves. All of the epithelial cells in all parts of the body. You can also touch your skin, yes, that you can touch all parts. Yes, it means included a lot of sensory cells or lots of sensory nerve fibers that have this function. Regeneration. Another fact in epithelial tissue. Again, in all parts, some cells are named as basal cells. These cells are similar to stem cells. Yesterday, I talked about that. The stem cells have two main characteristics. Their mitosis in the epithelial tissue, these cells again are in all parts. We now took a specific and main property in the connective tissue, that all of these cells have basal cells for regeneration, for renewal of the epithelial cells, and the second one, under them there are some type of connective tissue that give to them the nutrient. And finally, the special property of these cells is polarity. And we want to now about this in more detail. Polarity means they have different surface with different characterization. Yes? For example, these pointer have polarity. Here we have a brand, or this part don't have any. Or for example, have some part that I can use it. In the epithelial tissue, as this, that have some surface. Here is the lumen. Here another cell, another cell. This surface named as apical or free surface. That is toward the lumen. The opposite surface named as basal surface that located on lamina peripheria. And these are lateral, that by this surface they attach together. For the attachment to lateral, basal, or that are in the lumen for receiving some things, they have some structures that we want to know about them. Apical surface, basal surface, and lateral surface. Here, this picture shows the renal tubules, that the cells located adjust together the cell membrane, all of them located on the lamina peripheria and attached tightly together. But now we want to know about the structures that are in each surface, that these structures are useful or necessary for the function of the epithelial cells. The first surface is the basal surface. In your book, write it included two basal lamina and basement membrane. Do you know difference between of them? Mostly they may be used placed together. Basement membrane is here in the basal surface that attach the cell, epithelial cells to connective tissue. As you see here, they act as a supportive sheet between epithelium and underlying connective tissue. The basement membrane included basal lamina plus underlying reticular fiber. Thus, the basal lamina is a part of the basement membrane, but they use in different parts beside of the together. But both of these two structures, basal lamina and basement membrane, attach epithelium to underlying connective tissue and support it, prevent the rupturing or separation of the epithelial tissue from underlying connective tissue. Basal lamina mostly included some macromolecules. Lots of proteins, glycoproteins, protoglycan are in this part. That the most important of them is this collagen. Collagen, that we know about it in the connective tissue, until now have 28 types. That number as Greek number, 1, 2, 3, 4, on. That some of them well known and also are more important, and some of them just discovered and they scientists don't know any information about them. The type 4 collagen form a network in the basement membrane for anchoring and attachment of the other proteins to itself. That you can see their name and don't need to memorize them. Just now this one, because it's very, very important. The type 4 form a network, yes, schematically you can see here, is in the basal lamina, and also the basal lamina included the macromolecules. Macromolecules are proteins, lipids, carbohydrate, and also combination of these. For example, glycoproteins are combination of the carbohydrate and protein. But in the basement membrane, under the basal lamina, there is another layer that included the fibers. Which fibers? Reticular fibers. Reticular fibers are collagen type 3 that have another name as reticular fiber, because everywhere that included the collagen type 3, these collagen get the first one are very thin. The second one, they form a network of the fibers for the sitting, for the stroma of the connective tissue, for the sitting of the cell, location of the cells. This layer, this part included the reticular fibers or collagen type 3, named as reticular layer. The reticular layer plus basal lamina. Basal lamina, as you see here, just visible in electron microscope. We cannot see the basal lamina by light microscope, but with a special staining as PAS, we can see the basement membrane under microscope. But in the light microscope, with hematoxylin and eosin staining, we are unable to see the basement membrane. But we can imagine the location of it, because here show the epithelial tissue. The epithelial tissue included high amount of the cell. You can see a lot of nucleus. Yesterday, by hematoxylin, they have purple color, a purple spot. All of these spots, purple color, show the nucleus of the cells. That we can see a lot of them. But the connective tissue included few amount of the cells, mostly included a lot of the ground substance that under when we do staining, they don't like the dye. They don't have affinity to the dyes and have mostly white color. We can identify epithelial tissue from connective tissue or the lamina propria. Between of them, the structures is the basement membrane. In the basal surface of the epithelial cells, there is the basement membrane or basal lamina, and one of the junction that now about in the other slides. About the lamina propria, I thought. The lateral surface. These two surfaces located between of two cells. Two cells attached together by lateral surface. The first things that are in this surface are some structures that attach these two cells as a glue. That name the intracellular junctions, and also some proteins that link the cell together, named as cell adhesion molecule. Adhesion means the glue. This molecule acts as a glue and attach two cells together. Thus, in the basal surface, there are some structures for attachment of the cells. That these structures named as junctions. Cell tucked together, involved in the cell signaling of the cells. That now we want to know about in more detail. The first one. Functionally, we have three types. Yes, one, uploading. Second, adhesive or for attachment. And three, communication. Yes. The uploading junction again have just one type as tight junction or zonular uploaders. Uploaders means seal. Zonula means belt. Yes, they are as belt. Yes, that we put on in our pants. Yes, these cells also as a belt surrounded the peripheral part of the cells. It means the proteins that are in this junction encircle the cells are as zonula. Most apical. The tight junction always are here, the apical part of the lateral surface. Why? They prevent, they seal this part that the materials, anything, ions or others, prevent passing the materials from paracellular space. If the materials, any component want to enter, should pass from the cells. Where these junctions are? In the part of our body that included barrier, such as blood-brain barrier, you I think hear it. Yes? In the part that are included barrier. In the urinary bladder is a muscular sac that stores the urine. When urine formed and stored in the urinary bladder is a poison material for our body. It means it cannot come back to another organs or not to leach to underlying tissue. Included a lot of tight junction. Yes, the first one, occluding junction, have just one type as zonal adherent tight junction. It is most apical of the junction. Plasma membrane of the cell fused together as a zipper. Yes, as a zipper of your clothes. Yes, they attach, join together, point by point. There isn't any space or anything between of them, and also is as a belt. That its function is a prevention of passing anything. The main protein in this junction are clothing and uploading that express here and located between the cells and prevent passing of anything. The adhesion junction has three types as zonal adherent, desmosome, hemidesmosome. All of them are adherent junction are for the attachment of the cell together and also cell to the basement membrane, to the basal surface. The first one, zonal adherent, again is a belt-like structure, mostly located below the zonal uploaded here. But there are a space, for example here, and also they are as a belt. Between paracellular space, cytoplasmic surface of one cell, another cell. This cell attached together by some proteins that named as cadherin, glycoproteins. Cadherins are here in the paracellular space, and by some proteins, finally they attach to the actin filaments that are in the cell. The actin. Yes, this junction by some molecules that are adhesive junction attached to the actin filament. Yesterday, we know that all of the cells under the cell membrane included the actin filaments. There are some proteins that don't need to memorize them. Just to know that these cadherin protein in zonal adherent finally attach to the actin in the zonal adherent, attached to cell together. There is another type of the anchoring junction in lateral surface as desmosome. It is the macula adherent. It is as a plaque, as some the tight junction are as zip, the desmosome are as button. Yes, they are not in all part, just in some part they attach to membrane together. Here also in the paracellular space included the cadherin, but they in the desmosome that have plaque structures attached to the keratin. What is the keratin? Intermediate filaments of the epithelial cells. Cytokeratin or keratin. Intermediate filaments that are in the cytoplasmic part of two cells. They attach to the keratin. Hemidesmosome is in the basal surface, not in the lateral surface. It's hemi. It is the half of the desmosome. Desmosome have two plaques. One is one cell, the another one is the neighbor cell. But the hemidesmosome have one of it, have half of one plaque as this. Attach the cell to basement membrane, hemidesmosome. Hemidesmosome, as same as to desmosome, also attached to the keratin filaments or cytokeratin. But here, included the integrin beside of the cadherin that are here. Yes, integrin molecules attach this plaque to the basement membrane. Bond the cell to basal lamina or the basement membrane. Lateral surface adhesive junction. Just adhesive junction. In the adherent junction and desmosome in the paracellular space included the cadherins. The zonal adherent attached to actin filaments, the desmosome attached to intermediate filaments. And you know, in the epithelial tissue we have just keratin or cytokeratin as intermediate filament. The cell type as a communicating junction connected to cells together. Gap junction, another name of it. Located everywhere of the lateral surface because it wants to connect together. The cells want to exchange some things together as some small molecules or hormones. For this connection and communication, exchanging of some things, there is one structure that named connexon, this orange color. Yes, this is one connexon. Another one. In each connexon, there is 6 proteins as connexin that located round together and form very narrow canal that cells communicate together. They have connection together, named as gap or communicating junction, that is in the lateral surface. The lateral surface gap junction, one complex as a connexin that, the connexon that included the connexin proteins. 6 connexin proteins form just one connexon. This is a hexameric complex. Briefly show the surface, junction in the lateral surface. Occluding junction or tight junction. 2 cell membrane joined or attached together point by point. You cannot separate from them. Still, their paracellular space. Yes, as you see here, there aren't any spaces between them. Anchoring junction as desmosome, tupelage into adjust cells that included the adhesive glucose, intercellular space that they attach together by adhering glucose. In the zonal adherent that here act as a belt and are same to belt. The gap junction are everywhere and 2 cells connected together in the lateral surface. Any question? I continue or I think I tell the apical surface and then we have a rest. On the apical surface are some structures that increase cell surface area or move something. The first one, microvilli, are projection of the cell membrane in the apical surface. They increase the surface. Are in the absorptive or reabsorptive organs, are more in the small intestine, in the renal tubules. Yes, in the renal tubules, some materials reabsorb from the urine, the useful material. Microvilli are finger-like extension of plasma membrane. It means they included the cell membrane. If you memorize again, and also you now, under the cell membrane of the cells included the actin filament. When this membrane projected, bring for it by itself the actin filaments. Thus the main structure of the microfilament, the core of them, included the actin filament that's covered by the cell membrane. Increase the surface area in the small intestine or others. That here you can see them by special staining electron microscope. Yes, the structure of the microvilli composed of actin filament. You know why? Because actin filament are under the cell membrane. When the cell membrane projects, bring with itself the actin filament also. Cilia, yesterday we talked about that. That are some included the microtubules as 9 double tubuli plus 2 single in the center that are in the apical surface of some cells. They included 1 motor proteins as dynein, ciliary dynein. Again, yesterday we talked about that move mucus or another materials in the surface of the cells. This type of epithelium are in the respiratory system, from the upper part of the nose until the trachea and its branching. Also named in the epithelial tissue, we have some columnar ciliated cells because they included a lot of them. The cilia are white-like mobile extension, included the microtubules, are the respiratory tubes, move the mucus or other things that are longer and thicker than microvilli. The third structures in the apical surface are stereocilia. Stereocilia is a round form in the histology. Yes, this is the microvilli, the cilia. Stereocilia also are some extension of the cell membrane. Thus, structurally they are as two microvilli. Yes, included the actin, not tubulin. But why we name them as stereocilia? Stereo means immotile cilia. Because when these structures discovered in the cell, the electron microscope not discovered. And scientists believed because of its length, because it is not as long as the microvilli, but it's close to the cilia. They think that this is a cilia, but it's not. But after the discovery of the electron microscope, scientists saw that this is a projection of the cell membrane and also included the actin filament, not the tubulin proteins. Act as increasing of the surface of the cells. The stereocilia are where? In the epididymis and vasodephron of the male genital system. It helps to movement and maturation of the spermatozoa in the male reproductive system. Thus, in the apical surface, we have three structures as stereocilia, microvilli, and cilia that you can here compare the height of them, that the microvilli are short, the cilia are a little longer than microvilli, and finally the stereocilia is the longest one. The microvilli and stereocilia structurally are same. Both of them are projection of the cell membrane, included the actin filaments. But the cilia are motile structures that located on apical surface and included the tubulin. Have a 9+2 microtubules structure. And the lateral surface, the junction in the lateral surface that attach the cell together, and finally the basal surface. These specific characterization are in the epithelial tissue, but it not means that they are not in other tissues. For example, in the nervous tissue and isthmus muscle, there are a lot of gap junction. The first one. And the second one, it not means that all of these junction and all of these characterization are in one cell. No. Based on the function of the cells, maybe some of them are and some of them are not. For example, as I said for you, the tight junction are just in some cells, that the cells can exchange something together, don't have the tight junction.